Hello and welcome dear viewers to the another topic on quality control test for tablets. In earlier video we have seen the in process quality control test for tablet, and this video is regarding the finished product quality control test for tablets. This also includes the stability testing for the tablet formulation. So finished product tests are the tests which are performed after the process is completed, and these tests are performed in QC, and after completion of the test, COA, that is certificate of analysis, is generated, and based on that certificate of analysis the product can be released into the market. These tests are performed on the finished product. That's why these are known as the FP test or finished product test. The tablet formulation may be coated, uncoated, and maybe IR, ER, or maybe delayed release. So these tests are common for the formulations. Mainly disintegration test is not performed on the extended release formulations or sustained release formulations or modified release formulations. But these are performed for the IR and DR formulations. The reason behind not performing the disintegration testing for extended release tablets might be, or maybe, that these are the formulations, or these are the tablet formulations which are not meant to be disintegrated, and the disintegration time is very long for the extended release type of tablets. That's why generally DT test is not performed. Then the finished product test include the testing for the finished formulation for release of the batch into the market, and also for stability testing in the stability intervals. If you have made the batch and you are charging the batch on the stability testing, then these tests are performed for the stability intervals, so that the product behavior can be understood at different storage conditions, and also the stability is performed onto the formulations which are yet to be approved, like exhibit batches are there for which the stability testing is done. Then what are the quality control tests? So these are including the description, that is appearance or visual identity, tablet weight variation test, uniformity of weight or uniformity of mass, then identification. Identification is by IR, HPLC, and color identification. Uniformity of dosage units, so this is called as content uniformity. Then dissolution, then assay, related substances. Related substances are also known as impurity testing. Then residual solvent testing. If the solvents are used into the manufacturing, then residual solvent are required to be tested. And the microbial testing. Then other quality control tests are water content. Water content is evaluated by water by KF testing, that is Karl Fischer reagent is used in that testing. That's why it is generally known as water by KF. Then hardness, that is resistance to crushing. Then dissolution profile. Dissolution single point can be done, or dissolution profile can be done. Generally dissolution profile is done on the stability samples. Then disintegration time, that is DT. Then chiral purity or enantiomeric purity. If the API contains the chiral impurities or enantiomeric impurities, then this test is performed. Then description. So first is description, that is the visual appearance of the formulation. Tablet description appearance testing is not an official test, and it varies from product to product. For example, yellow round uncoated or film coated tablets with L debossed on one side and 5 on the other side, then it should be free from the physical defects. If the formulation has the break line, then break line details should be included. Description is required to identify the product. It is required for patient acceptance. and it also gives you the information regarding problem-free manufacturing and batch-to-batch uniformity. Also, the manufacturing defects can be understood from this visual description test, whether there is chipping, double embossing, dragging, sticking, picking, rough surface, or any coating defect, shade variation. So these are the important things which can be understood from the description test. Then is the weight variation test. So weight variation test is done as per the compendial requirements from IPBP or USP, and in this individual tablet weight and the group weight is taken, and based on the tablet weight the percent deviation is allowed. So maximum percent differences allowed are as per the European Pharmacopoeia, Indian Pharmacopoeia or British Pharmacopoeia. So these are generally 5%, 7.5% and 10% from the mean value. If the tablets are outside the range specified, then the weight variation test fails. Then hardness, it is also called as resistance to crushing, and mainly in the European Union, this hardness is known as resistance to crushing. So tablet hardness is not an official test, and it depends on to the product, and this is the test which ensures that the tablet can withstand the mechanical pressure during manufacturing processes like coating, packaging, storing and handling. And also, if there is a change in the hardness of the formulation during stability, then that can be indicated by the hardness test. See, this hardness test has the impact on the disintegration, and disintegration has impact on to the dissolution. That's why hardness test is also done uh, for the finished product and during the stability studies. Hardness is finalized based on the scale-up trials and R&D trials, and generally 10 tablets are used to test the hardness. Monsanto hardness tester, Pfizer hardness tester, and nowadays automated hardness thickness testers are there to perform the hardness test. Then DT testing. This is called as disintegration test, and as per the pharmacopoeial requirements, the DT time is given. So for uncoated tablet, generally 15 minutes is given, and if the formulation is film coated, then 30-minute time is given. So based on the formulation design, this DT time is different. Like sugar coated tablets has the DT limit of 60 minutes as per British Pharmacopoeia. Generally 6 tablets are used, and if one or two of the 6 tablets fails in the DT test, then additional 12 tablets are tested, and if the requirement is meeting, that means not fewer than 16 of the total 18 tablets are disintegrated, then the formulation passes the DT test. Then coming to the identification. So identification is required to be done by different principles like UV and non-UV. So HPLC and IR is done. And for the European Union, color identification is required. So if the formulation contains the color and titanium dioxide, that time this color identification test is required. So in HPLC testing, retention time of standard is tested and compared with the test sample. And similarly in infrared spectra, the infrared spectra of test should match with the standard spectrum. Then uniformity of dosage units. This is also called as UOD. Earlier it was done by the mass variation, that is uniformity of dosage units by mass variation or weight variation. But nowadays all the regulatory authorities are asking for the uh, uniformity of dosage units by content uniformity. But earlier, if the percentage of the API content or API load in the formulation, if it is about 25% or 25 mg, then uh, content uniformity was not required. But nowadays, content uniformity is compulsory for all the formulations. Content uniformity AV value is required, and this AV value should be equal to or below 50. So if this criteria is met, then it passes the uniformity of dosage units. Dissolution. Generally Q point is tested for IR formulation. If the formulation is ER, then multiple time points are considered. For example, for IR formulation, not less than 80% Q dissolved in the 30 minutes. It is the criteria. And for MR formulation and DR formulation, different criteria is given as per the formulation design. Also, if the tablet is coated, that means if the tablet is with break line, then the dissolution of whole tablet and the half tablet is to be done, and separate testing is required onto the break or split part of the tablet or broken part of the scored tablet. Then assay is the percent of label claim or percent content of label claim. If the assay is not within the specified limits, then the formulation will fail. The label claim means whatever the label of the strength is given, that means 100 mg, each tablet contains 100 mg, that is the label claim. Each tablet contains or each capsule contains 100 mg of the, for example, paracetamol, and if it is out of the percentage limit, then it will fail, whether it is on lower side or whether it is on the higher side. So assay is very important. For European Union region, 95 to 105% is given, and for US generally 90 to 100%, 90 to 110% is given. So these are the generalized limit for assay. Then related substances. The related substances testing is also known as impurity testing, and in this mainly the degradation products are tested. So known impurities are tested, whether the impurity is known or unknown, but if it is degradant, then it is required to be tested. And some unknown impurities are also there, which are specified by RRT. So if any RRT based impurity is there, that is required to be tested. Then single max unknown impurity, or any, it is also known as any other individual impurity, that is unspecified. So out of the all unknown impurities, whichever is higher, that is required to be tested. And finally the total impurities are tested. Along with this, the enantiomeric impurities are tested. Then residual solvents. So if any solvent is used into the manufacturing, that time the finished formulation should be tested for the residual solvents. And as per the ICH guidelines, the limits are given at ppm level. So the example of residual solvents are isopropyl alcohol, MDC, acetone. Another testing is water content. It is done by KF. And the last is microbial testing. So for tablet formulation, generally microbial testing doesn't pose more challenges, but certainly microbial testing is very important testing. For solid oral formulations, microbial testing is done, and in microbial testing, total aerobic microbial count, that is 10 to the power 3 CFU, that is colony forming units, is the limit. Total combined yeast and molds. Then specific microorganisms include E. coli. This E. coli should be absent. In stability testing, generally, the sample stored at accelerated, intermediate, and long-term conditions are tested. And these samples may be in the different packagings like SDP bottle, blisters, and many others. So in stability testing, generally all the tests are required to be performed except the identification test and the residual solvent testing. So why these tests are not done in the stability testing? See, because once the formulation is loaded on stability, that formulation is tested for the identification, and identification does not change with time. If the tablet contains paracetamol and it is identified, then the same formulation will contain paracetamol and it is not required to be identified once more. Then residual solvent testing. So once you finish the process and you test the residual solvents by residual solvent testing, generally by GC, then there are no chance, or there are very less chance for variation in the residual solvent content. And in stability, the residual solvent may not get increase. That's why in stability testing these two tests are not done. So this is regarding the quality control testing of tablet formulation. Similar tests are there for capsule formulations as well. Only the hardness testing is not done on the capsules. So the same testing is done for capsule formulation as well. Also, soft gelatin capsules are tested in the same way. The assay, RS, DISO, and water content, these are the prime important tests for the oral solid formulations. So I hope this video will help you to understand the quality control test for tablets for release and the stability testing. Generally, release test or release testing means the test on finished formulations performed to release the batch into the market, and stability testing are the tests which are done onto the finished formulation and during the stability intervals to prove the stability of the formulation, and also to give the shelf life and storage recommendation for the formulation. And we will meet soon with such kind of informative video. Till then, take care and bye bye. Keep watching Pharma Learning in Depth channel videos. Thank you.